Hey guys, this is Mrs. Krause, and this is our second video on meiosis. So in our first video, we left off talking about crossing over between pairs of homologous chromosomes. Question number 6 in your notes on the right-hand side asks, how does crossing over contribute to genetic variety? And we're going to answer by saying that crossing over allows trading of genes, and that results in every egg that's created and every sperm that's created by the same person being genetically different from one another. So each egg that your mom made and each sperm that your dad made were different from each other, and that's why you and your siblings look different from each other and both of your parents. Okay, unless you're identical twins. Question number 7 asks, why is a pair of chromosomes called a tetrad? Tetra actually means 4, and if you think about it, within this pair, there are 4 pieces of DNA, 4 chromatids. One on the side of this chromosome, one on the side of that chromosome, one here, one here. So 4 chromatids total would be tetra. All right. Okay. Now we've talked about prophase 1 when the homologous chromosomes come together and exchange DNA. During metaphase 1, those pairs of chromosomes will line up at the center of the dividing cell, attached to the spindle fibers, but instead of lining up 1, 2, 3, 4 in a row, they're going to line up in pairs like you see in this picture. Okay. You are asked to, on your notes, label several things. Here it asks you to label the homologous chromosomes, the centromere, which is where the spindle fibers attach to the chromosomes, the spindle in yellow, and the metaphase plate. Okay. All right, our next step is anaphase 1, and during that step, our homologous chromosomes get pulled apart from each other. So at this point, we're not ripping apart chromatids like we did in mitosis. We're pulling apart these pairs of homologous chromosomes. Okay. For number 9, you're asked to label both the homologous chromosomes and the spindle. If you just want to label the homologous chromosomes, that's fine because we labeled the spindle in the previous picture. During telophase 1 and then cytokinesis, it's very similar to the telophase that happens in mitosis. In this stage, the chromosomes get to opposite ends of the dividing cell. The cytoplasm divides, and we are going to be rebuilding our nuclear membranes as well as our nucleoli, and the mitotic spindle is going to break down. Okay. We're going to end up with 2 daughter cells that have half the number of chromosomes as the parent cell. In this case, we had 4 chromosomes in our parent cell, and they were all replicated. They looked like X's. And now we end up with 2 daughter cells that have 2 chromosomes, and they are still replicated. They still look like X's. So we went from 4 chromosomes in the parent to 2 in the daughter cells. For number 10, we are asked to label the nuclear membrane, which we see as these little pieces. Oh, sorry, you can hear the baby crying in the background. These little pieces of white material that are forming around the chromosomes. You're also asked to label the centrioles. The centrioles, remember, we'll get in a better picture here. The centrioles are the little churros that are in the center of the centrosome, which kind of looks like a sun around it. So you could label both the centrioles and the centrosome in your picture. Okay. And it also asks you to label the chromosomes. Since we've already labeled them, you don't need to do that if you don't want to. Okay. And on to meiosis 2. Okay. Once we've finished with meiosis 1 and we have 2 daughter cells, each with half the number of chromosomes as the parent cell, we're going to enter meiosis 2. It's important to note that the DNA is not copied again. Okay. Now, question number 11 asks, when was the DNA copied? And remember, the DNA was originally copied during the S phase of interphase that took place before meiosis 1. During meiosis 2, we're going to see something very similar to what happens in mitosis happen, because instead of pulling apart pairs of homologous chromosomes like we do in meiosis 1, in meiosis 2, we're going to be separating chromatids, identical copies of DNA. So we're going to be ripping apart the pieces of that X, just like we do in mitosis. We have 4 stages. They're the same ones as we see in meiosis 1 and mitosis, but this time we put a 2 after each one. Okay. Okay, so this picture is just taking both of the daughter cells of meiosis 1 on the left. They're going to enter their own prophase 2, and they're going to build their mitotic spindle, break down their nuclear membrane and their nucleolus. And we are going to, during metaphase 2, move the chromosomes in each daughter cell to the center of the dividing cell. Now, this time, they're not going to line up in pairs. They're going to line up in one long line, single file, like this. So then in anaphase 2, we're going to be separating the chromatids from one another, and sometimes we call the chromatids sister chromatids. Once we separate the sister chromatids, they're considered daughter chromosomes. And then in telophase 2, we can see that our DNA is starting to unwind back into chromatin. But if we could see it still as chromosomes, we would see that we have 2 chromosomes in each of our daughter cells. And this time, since we've separated the chromatids, they are not replicated. They don't look like X's. So we have 4 daughter cells here. Each one has 1, 2, 1, 2, 1, 2, and 1, 2 chromosomes. So just to kind of summarize that, question number 12 asks, how is the end result of meiosis different from the end result of mitosis? And we're going to say the end result of meiosis is going to be 4 haploid daughter cells, whereas the end result of mitosis is 2 diploid daughter cells. We're calling these 4 daughter cells haploid because they have one set of chromosomes. In this case, one set of chromosomes is 2 chromosomes, whereas diploid would be 4. And in human cells, one set of chromosomes is 23, and 2 sets would be 46. So each of these 4 daughter cells in humans would have 23 chromosomes. Okay, now we're going to label a couple things in our pictures of the stages of meiosis 2. We don't need to label anything in this first picture, even though it says 2 and number 13 on your packet. We've already labeled the spindle, the nuclear membrane, and the chromosomes in previous pictures. For metaphase 2, I think it might be helpful to label the chromatids, so the 2 sides of the X, and also to label the centromere where the chromatids join to each other, and that's also where the spindle fibers attach to the chromosomes. Okay. And then in anaphase 2, we're going to label the new chromosomes, and I might also write daughter chromosomes as well, because that's what we've called them in the past. So once the chromatids separate from each other, they're considered daughter chromosomes. Okay. And then you may want to label in telophase 2 and cytokinesis that our chromosomes are uncoiling into chromatin. That's why they look so long. But you can still count 1, 2 in each cell. 1, 2. Okay. Now our overall goal, as we mentioned before, was to create sex cells. And another term for sex cells is gametes. Gametes are defined as haploid reproductive cells that we make during meiosis, so sperm and eggs. And also, as we mentioned before, sperm are made in the testes in humans, and eggs are made in the ovaries in humans. Okay, and this is just showing an actual picture from an electron microscope, I believe, of a sperm fertilizing an egg, and it actually uses enzymes in the head of the sperm to break through the egg's outer layer. Okay. Now, there are a couple differences between meiosis to create sperm in males and meiosis to create eggs in females. We call meiosis to create sperm spermatogenesis, and that makes sense because genesis means to make, so we're making sperm. And we have a diploid parent cell in the testes that's going to divide by meiosis to create 4 haploid sperm cells that are all totally functional. And this is actually an image of sperm being created in the testes. So I think each one of these purple dots is the head of a sperm cell that's being created. This diagram just shows that through meiosis 1 and meiosis 2, we end up with 4 functional sperm cells. Oogenesis, and it is pronounced oogenesis, not oogenesis, is the process of making eggs. I think they call it oogenesis because an egg cell is technically called an ovum, and genesis means to make. So we're making an ovum or an egg. So unlike spermatogenesis, we have a diploid cell that's in the ovaries that's dividing by meiosis, but we end up only making 1 mature egg cell. As we can see in this picture, we have our first round of meiosis, and cytokinesis, the division of the cytoplasm, happens a little bit unevenly during the first round of meiosis, meiosis 1, so that this daughter cell gets a little more cytoplasm than the other. They still have the same amount of DNA. Okay. Then during meiosis 2, when these 2 cells are dividing to make 4 daughter cells, cytokinesis happens unevenly again. So this one cell, the egg cell, ends up getting most of the cytoplasm. And that's because when you're creating a fertilized egg that's eventually going to divide multiple times and create a baby, the egg is what brings the cytoplasm and all of the organelles to create that fertilized egg. The sperm really only brings DNA. That's about it. Okay, so we have this one egg cell that's created, and then we have these 3 tiny cells. And these 3 tiny cells are called polar bodies. Number 17 asks us to describe how the polar bodies are different from the egg cell. They have much less cytoplasm and organelles than the egg cell does. And again, that's so the egg cell can bring enough nutrients to the fertilized egg or zygote. And eventually our polar bodies disintegrate and die, even though they're carrying the same amount of DNA as the egg. Okay. Number 18 in your notes asks, what's the main difference between spermatogenesis and oogenesis? And we would describe the main difference as spermatogenesis makes 4 functional sperm cells, and oogenesis is only going to create 1 functional egg cell for each parent cell. Right. In your notes, you have a diagram that compares meiosis on the left with mitosis on the right. And really all that they're showing in the meiosis picture is primarily meiosis 1, because we're seeing the homologous chromosomes line up in pairs, they're splitting, and then these would be the daughter cells of meiosis 1. And they don't show meiosis 2 at all. They just show the end result of meiosis 2 with our 4 daughter cells. Okay, but if we were to show a picture between our 2 daughter cells of meiosis 1 and our 4 daughter cells of meiosis 2, we would show these X-shaped chromosomes lining up single file in a line at the center of the cell, and then the chromatids separating from each other. Once the chromatids are separate, we call them daughter chromosomes, and we end up with 2 daughter chromosomes in each of our daughter cells.